In today's wee little video on metabolic health, we are going over ketogenesis and a teeny little bit about starvation. So today's little agenda, we're gonna discuss what the frick even are ketones, how ketones are made by acetyl-CoA in your liver, how ketones give your brain so much energy, and then we're gonna discuss in what cases ketones are actually gonna be produced by your liver. So to start this little video off, what even are ketones? Basically, your body has 3 ketone bodies that it forms, and these are all being formed in your liver by excess acetyl-CoA. And we're going to talk about how you get to excess acetyl-CoA in 1 minute, but these are your primary ketone bodies: acetone, acetoacetate, and betahydroxybutyrate. A lot of you guys have definitely probably already heard about betahydroxybutyrate or BHB because it is such a potent fuel molecule for your brain. This is like rocket fuel for your brain. Acetone is a little useless. You will literally just like breathe it out of your lungs. It's super tiny and it's very volatile. So as soon as your liver starts pumping out acetone, that acetone escapes your liver, ends up in your blood, and if it circulates to your lungs, you're literally just gonna exhale it. So this is why like sometimes people on keto diets, their breath might smell like nail polish remover. It is literally because acetone is nail polish remover and they are breathing out acetone. It's volatile, you just lose it from your body through your lungs or through your sweat. Beta-hydroxybutyrate, you are keeping this in your body, and when your liver makes beta-hydroxybutyrate, it pops that out into your blood, and now it's gonna circulate to your brain and it's just gonna light your brain up. So let's figure out how the frick ketones are actually made. So ketogenesis is the creation of ketones. Ketogenesis, making ketones. Ketogenesis happens in your liver, and its substrate, its starting molecule, is acetyl-CoA. We need acetyl-CoA to make ketones. And these acetyl-CoA molecules come from beta oxidation of fatty acids. So if you've got a good grip on beta oxidation, you know that the end product is acetyl-CoA. When we burn fat, we turn the fat into acetyl-CoA, and then acetyl-CoA can enter the tricarboxylic acid cycle, and that's when we get ATP or energy from burning the fatty acid. If you have high rates of beta oxidation, you have so many fatty acids flooding your liver and you are making so much acetyl-CoA, not all of that acetyl-CoA is gonna be able to be processed by the tricarboxylic acid cycle. When the acetyl-CoA pool is greater than what the tricarboxylic acid cycle can process, we will send acetyl-CoA down ketogenesis. So this is the actual ketogenesis pathway that your liver is sending acetyl-CoA down. Up here, we start with 2 acetyl-CoA molecules, and we use the enzyme thiolase to hook these molecules up and spit out CoAsh. And spitting out this CoAsh here is super important because it's gonna help us keep on doing beta oxidation. It's gonna help us burn more fat and make more acetyl-CoA. So thiolase unites acetyl-CoA, spits out CoAsh, and this gives us acetoacetyl-CoA. The next enzyme is HMG-CoA synthase, and it uses acetyl-CoA and water to convert acetoacetyl-CoA into HMG-CoA. So HMG-CoA looks like this, and we're a teeny little bit closer to making ketones, and now we have to use the enzyme HMG-CoA lyase to pop off an acetyl-CoA. So this part right here is acetyl-CoA. HMG-CoA lyase chops right here, and we remove this part of the molecule and we spit out acetyl-CoA. Now we have acetoacetate. And acetoacetate, this is one of our ketones, but we're not like quite finished the complete process of ketogenesis. Our goal is to get beta-hydroxybutyrate because beta-hydroxybutyrate, this is like that rocket fuel for your brain. This is what we really want. So acetoacetate at this point has 2 fates. We're gonna use the enzyme over here, acetoacetate decarboxylase, to remove the equivalent of a CO2 molecule, so COO, C, 2 oxygens, and then spit out acetone. So what that kind of looks like here is we chop this part off, COO, whoosh, see you later, and we keep this part, acetone. So it gets chopped in half, we get rid of carbon dioxide, you breathe that out, and we've generated acetone. Acetone is gonna hop on out of your liver, hop into your circulation, and it's just gonna go woo all around your body until it gets to your lungs and you exhale it. On the other side, we have beta-hydroxybutyrate being made, and this is where we're gonna use the enzyme beta-hydroxybutyrate dehydrogenase, beta-HBDH, and we're gonna use NADH to reduce acetate and yield beta-hydroxybutyrate. So look at the changes that we've made. NADH reduces this carbonyl group right here, and it gives us an alcohol group. So we change right here into an alcohol group, and we make beta-hydroxybutyrate, and then we also spit out NAD+. This beta-hydroxybutyrate is literally gold for your body. Your liver spits it out into your blood, and it's gonna travel all around everywhere, and it's gonna enter your brain, and now your brain can run on ketones. Now this beta-hydroxybutyrate is gonna serve as a very, very rapidly available, potent source of energy for your neurons, and it's just gonna help accelerate your brain function. So when beta-hydroxybutyrate actually shows up in your brain, this little pathway is how we get the energy out of beta-hydroxybutyrate. Remember that we made these ketones from acetyl-CoA? Acetyl-CoA is the entry point to the tricarboxylic acid cycle. When you are burning sugar for energy, sugar goes down glycolysis, then you make pyruvate. Pyruvate goes through the pyruvate dehydrogenase complex, then you make acetyl-CoA, and then acetyl-CoA enters the tricarboxylic acid cycle, and you just pump out so much energy. Same thing with fatty acids. When you are burning fatty acids, when you are sending fat down beta oxidation to make energy, you go fatty acid, acetyl-CoA, acetyl-CoA hops in the tricarboxylic acid cycle, and you spit out energy. Beta-hydroxybutyrate also is gonna be processed into acetyl-CoA inside of your brain, and that acetyl-CoA is gonna give you the energy. So let's zoom in on this pathway. Here we start with beta-hydroxybutyrate, and we use the same enzyme, beta-hydroxybutyrate dehydrogenase, to yield acetoacetate. So this reaction is reversible, and it will either make beta-hydroxybutyrate or it will make acetoacetate. So we use NAD+ to oxidize beta-hydroxybutyrate and yield acetoacetate. Once we have acetoacetate, we are going to use succinyl-CoA and the enzyme beta-ketoacidyl-CoA transferase to make acetoacetyl-CoA. So succinyl-CoA comes in and we spit out succinate. Acetoacetate comes in and we spit out acetoacetyl-CoA. Acetoacetyl-CoA then enters the enzyme thiolase, and just like as we saw in the liver in ketogenesis, thiolase is a reversible reaction, so all we're doing here is just running it the opposite way. So that's ketogenesis, and that's how your brain uses ketones for fuel. When are we actually making ketones? Like, where does this happen and why does it happen? Basically, the only time you're gonna have significant amounts of ketones being produced, or your body's gonna be in like a keto state, in ketosis, is in starvation or in extreme carbohydrate restricted diets. And this means like restricting total calories from carbohydrate to less than 5%. If carbohydrates start creeping up, or if protein starts keeping up, that is literally just gonna kick you out of ketosis. In starvation, your body's gonna really start tapping into its stored body fat as a source of energy. So now all of a sudden you've entered the state of starvation, metabolic starvation, and now huge amounts of fats are being released from your fat cells. All these fats are entering your blood and they are traveling to your organs for oxidation as a source of energy, but your brain does not do a very good job at burning fat. And because we are in starvation, there is very, very low glucose, and we are also not really doing gluconeogenesis from amino acids, because in this state of starvation, we need to spare our body proteins. Like, we cannot just start burning amino acids in our liver or in our kidney or in our heart for energy. That's literally just gonna kill you. We have to rely on these fatty acids as a source of energy. And then in the case of a keto diet, when you are eating just so much fat and so little carbohydrate, you are gonna be doing so much beta oxidation of these fats in your liver, and you're gonna be making so much acetyl-CoA. So in the case of starvation, in the case of a keto diet, we have huge amounts of fats showing up in your liver, and we're gonna oxidize those into acetyl-CoA. And something super interesting in metabolism that is happening when we are in starvation is we're gonna start to deplete the TCA cycle of its intermediates. So in the late fasting state, we're not in starvation yet, we're still in a long fast, you are gonna be burning some amino acids for energy. You have high glucose demands. What your body's gonna do is it's gonna start breaking down body proteins, and it's gonna send those amino acids into gluconeogenesis. And then some of these TCA cycle intermediates are gonna contribute to gluconeogenesis. So we're gonna pull oxaloacetate out of the TCA cycle and process that into glucose, and now blood glucose is stored. But this process is starting with amino acids that we got from body proteins. Once we transition from fasting state into starvation state, your body's gonna start releasing huge amounts of fats into the blood, and it's gonna put the brakes on protein catabolism. It's gonna stop breaking down body proteins. And the reasons why it's doing this is because at this point, your body understands, like, no food is coming in, there's no sugar, no fat, no protein. We are literally starving. I need to save my organs. I cannot break down my liver. I cannot break down my cardiac tissue. I cannot break down my organs as a source of energy. I have to preserve them to preserve body functions and literally survive. So as soon as we shut off this gluconeogenesis from amino acids, we're still pulling oxaloacetate out of the TCA cycle to make a little bit of glucose, but we're not replenishing the TCA cycle. We start depleting the TCA cycle of oxaloacetate and all the molecules critical to actually have it spin around and function. When we deplete the TCA cycle of its intermediates, we can't keep sending acetyl-CoA in there, but we keep on burning fat. Our fat cells keep on dumping lipids into the blood. Those lipids keep on showing up into the liver. They enter beta oxidation. They give us acetyl-CoA. Now acetyl-CoA has nowhere to go. This is just gonna step the gas, this is gonna crank up ketogenesis. The acetyl-CoA enters ketogenesis, and we will make more and more and more ketones. That is what's going on in starvation when you have huge rates of fat oxidation and you begin entering ketosis. And long-term ketosis can cause a lot of damage to your organs, to your tissues, because ketones are keto acids. So if you have high amounts of circulating ketones, these ketones can acidify your blood and cause damage to your organs. So obviously starvation isn't a good thing. You are going to be starving until at some point you die, and that is because you have no energy, your organs are falling apart, and your organs are being damaged by the amount of acid accumulating in your blood. Okay, this concludes today's wee little video on ketosis. If you guys got any value out of this video, like and subscribe. You have no idea how much it helps me grow this channel and put more biochemistry, metabolic health, educational content out there for you. And then most of my videos are accompanied by some sort of note package. You can go to my website, nutritionbyelsa.com, where you can see the notes for most of my videos, basically all of them. And I hope you guys have such a good day. Goodbye. Mwah.